Warm greetings from San Diego to Japan Human Brain Mapping participants. In this presentation, I'll show how to use EMSE, or EMSE as I usually say it, to conduct spatial component analysis of a multi-condition group event-related potential experiment. Here's the plan. We'll start by summarizing the experimental design of the sample data set. This comprises 128 channel EEG data for 13 persons and 6 experimental conditions. As we'll see, there are 2 tasks. One task had 2 conditions and the other task had 4 conditions. But they used the same stimulus codes. Next, we'll move to spatial component analysis as a channel reduction technique. A way to map actual EEG channels to a smaller number of virtual channels. In this example, we'll reduce 128 EEG channels to 4 virtual channels. We'll use a particular kind of spatial component analysis called Infomax Rotated Spatial PCA, which is equivalent to an application of Infomax ICA for ERPs. There are 4 main steps in this analysis, and in the interest of time, we'll have to pass very rapidly through the first 2 steps. Let me just note here that in step 1 we start with the raw EEG. We can use a script to generate Workspace and perform some filtering. Then in step 3 we can do a visual review of the data and apply spatial interpolation if necessary and perform ocular correction. In step 3 we'll perform ERP selective averaging and group averaging using the event pipeline in EMSE. Show that in as much detail as time permits. And then finally, in step 4, we'll perform the Infomax Rotated Spatial PCA of concatenated group average ERPs that were generated by the event pipeline. So it'll be trained in essence on the group averages, but it will get applied to all the individual ERPs. In practice, there would be another step, which would be to perform some statistical analysis such as ANOVA, but this step is not covered here. Okay, so let's jump into the experiment. Back when EMSE's event pipeline had just been developed, Dr. Kirk Daffner, who directs the Laboratory of Healthy Cognitive Aging in Boston, provided this very nice data set in order to test the whole procedure. This paper describes the experiment. Aiming to study ERP effects related to executive functioning, the researchers devised 2 different continuous performance tasks for the same sequences as illustrated here. Letters were presented randomly with one of 2 colors, red and blue. In the neutral task or neutral condition, the participant's job was to press a button when one of 5 designated target letters appeared. The probability of a target was 15%, and so the probability of a standard non-target was 85%. So in this neutral condition, color doesn't matter, it was irrelevant to the task. In the selective attention task, the participant had to respond only to targets of a designated color, that is either red or blue, as specified at the beginning of the run. Therefore, the participant had to ignore or suppress target-like distractors which had the unintended color. So here, for example, if the intended color is red, and the letter B is not in the target class, then this would be a standard in the intended channel. If A is not in the target set, then this would be a standard in the ignored color channel. Here, if T is one of the target characters, and it's red, so this would be a target. Respond to this one. This would be here a target like distractor that the participant would need to suppress or ignore responding to this case. So the neutral task has two kinds of events, standards and targets. The selective attention task has four kinds of events, targets which have the attended color, standards which have the attended color, target like distractors with the unattended color, standards of the unattended color, for a total of 6 event-related experimental conditions. Here we see the 128 channel layout and also 10 channel groups that were of interest to the researchers. Channel group averaging is a common a priori way to reduce the dimension of a dense EEG dataset. EMSE has a channel group averaging filter for this purpose. However, of course, this procedure does not make the best use of all 128 channels. But a spatial component analysis is a data-driven way to reduce the dimension. It uses all available channels to explain observed variance across the experimental conditions of interest and across a specified time interval of interest. As you know, spatial component analysis, or spatial PCA, is a straightforward data reduction technique which finds the fewest number of components to preserve a given percentage of variance of the data. However, each component is orthogonal with all the others, and this is not a natural physiological constraint. Consequently, the number of data space dimensions is reduced, but each individual dimension may not have physiological meaning. Following the use of spatial PCA to reduce the data space dimension, PROMAX and INFOMAX are both principled ways to relax the orthogonality constraint by rotating the original PCA dimensions. In this simulation study, Joe Dean and colleagues concluded that INFOMAX is the most effective for spatial analysis. They also concluded that PROMAX was most effective for temporal analysis. I'll argue in my English session talk that spatial analysis is what we want to do for EEG and MEG. So note that INFOMAX is precisely the same criterion that's used in the popular method of INFOMAX independent component analysis. Spatial ICA has many other uses as well as the one we'll use here, but here we're using it as a way to achieve physiologically meaningful components after a spatial PCA step to reduce the dimension of multi-condition group ERPs. So, therefore, as we've noted already, the ERP component approach we take here may be called spatial PCA with INFOMAX rotations. Okay, so let's jump ahead to the final result so we know what we're aiming for. Here we have the spatial topographies of 4 INFOMAX rotated spatial PCA components that account for 98% of the variance of the grand averages for all 6 conditions in the latency range from 0 to 800 milliseconds. Note that the variance accounted for here is spatial across all 128 channels. It's temporal because we did the analysis from 0 to 800 milliseconds. And importantly, variance is experimental. That is across the 6 experimental conditions. So the experimental design makes an important difference here. This analysis was made on grand averages, so these results do not take the variance across individual subjects into account. Okay, so the top row shows the flat map topographies for the floor components. These are normalized so there's no scale. Also, as we'll discuss, the polarities, positive and negative, are arbitrary at this stage. EMSE labels these ICA1, ICA2, ICA3, and ICA4. Now, in the bottom row, we're looking at 3D views of the same topographies using the EMSE Visualizer module. I've given them labels that are intended to have some physiological significance. We get to this at the very end. We'll see if my tentative labels make some sense. But let's emphasize again that the polarity of a spatial component can come out either way, positive or negative in any location, because the spatial topography must be multiplied with the component time series to get the true polarity. For example, this component, which I'm tentatively calling P3B, has the right spatial distribution for a P3B, and indeed it's parietal positive in the 300 millisecond latency range, because the topography and the time series are both negative. So negative times negative equals positive. We'll see that when we get to the end. Okay, let's get back to the original data. So I briefly showed a step 1 script that was used to generate Workspace. And in this EMSE Workspace, we're organizing it with each project is a participant. So for example, here we have a project participant 12JC. And here we're looking at a time series. And if we look at the event pipeline here, we'll see the filters that have already been applied. So we automatically applied a digital reference montage, which is an average mastoid here. We applied a polynomiality trend, third order polynomiality trend over the whole data file. And we did a Butterworth IIR filter from 0.03 to 40 Hertz, filter order 2, and importantly, this filter has already been applied to the whole data set. So that's nice. We don't have to do it. It speeds things up, but it also gets rid of any warm-up artifacts. The warm-up artifacts are at the end of the data. Now, in step 2, we visually review the data. We might have had a spatial interpolation filter. See in EMSE, if we needed to put a spatial interpolation filter, that would be here. Any bad channels we could specify, and if there aren't too many of them, we can do a nice job of restoring the bad channels. But this one didn't have that, but we did use an express ocular artifact filter. Let's uncheck this one. I'll say OK. Okay, so here we see a blink that was nicely corrected by this express ocular artifact correction filter. It turns out this filter worked well, very well for this data set for all the subjects. That's not always the case. Maybe because we had 128 channels might have been one reason why the express method works so well. If the express method doesn't work, and you should always check it out, don't just apply it blindly, then there are other methods that you can use to correct ocular artifacts. Okay, after step 2, we didn't yet have these averages here. We only had these two. And we didn't have grand averages either. So how would we do these averages? Let me take just a little bit of time to show you how this is done. We use the event pipeline at EMSE. Okay, so I'll go to Batch, Event Pipeline, and in this case we actually had 2 event pipelines, one for each of the conditions. So let's look at the more complicated one, the spatial attention task, the pipeline for that. And in step 1 of the pipeline, we need to give just an example of a data set, any data set that has all the events of interest in it as they were coded in the experiment. Then in step 2 here, we defined a factor which has these 4 cases in it. So standard attend, standard ignore, target attend, target ignore. Okay, we're going to use that in step 3. Okay, so this has already been done. This is very, very flexible for doing conditional averaging. The details of this depend, however, on the details of your coding of the events in your experiment. But let me just indicate how this is done. So in the standard ignore, there's going to be a tag, standard ignore, and targets. Okay, this is potentially confusing. Targets here, in this context, has nothing to do with the targets in the task. Rather, these are the event targets that we want to average with respect to, so the time 0 will be 0 on these events. Okay, so in this case, the standard attend case, we selected all these event codes based on the details of the experiment. They were standard attend. And then we defined so those are shown here as this 1, T0. Now we have one conditional. We have one conditional. And the conditional in this case was to a correct response. In this case, the correct response was no response, but the experimental control program knew that and put out this event code 5 when they correctly did not respond. Okay, so we're looking only for the trials where the participant performed correctly. And this is a conditional. And you can have conditionals on conditionals on conditionals if you need them. So this is very flexible. Okay, let me cancel here, go to the bins. Okay, so now here we're going to define, in this case, 1 bin per tag. And if we review them, so in this case there's a 1-to-1 correspondence. It doesn't always have to be. But these bins are the ones we want to make averages for. Standard attend, standard ignore, target attend, target ignore, and they're all sharing the same epoch specification here. So we made epochs from 200 milliseconds to 1,000 or milliseconds or 1 second. We didn't need to specify a filtering interval option because we pre-filtered the whole data. So we don't need to worry about warm-up artifacts, same as interval of interest. We want to do baseline correction in the 200 millisecond interval, and we want to do some thresholding, rejection. We want to do some rejection if they exceed thresholds. Okay, and all those criteria get applied to each of these bins. Okay, so cancel there. Next, we select the time series. So here we have a list. All the ones checked here are for the spatial attention task. Okay, those are the ones we want to make averages for here. Let me skip to the output where we're doing time domain averages, not power spectral density, coherence, or time frequency. And we're going to make grand averages. We're going to create this new project called Grand. We're going to put them there. Okay. If we say execute, it goes through all the participants, creates these averages, and makes the grand average folder here. And I'm not going to do it now. It's already been done. But it's automated, and the averages are given names automatically. The same thing, we had another pipeline for the neutral condition. Let me just show you how I might do that. I would go to load all, and there's another pipeline for the neutral case, has the parameters. They're all here. Just to look at the bins, now here in this case we only have two averaging bins, standard and target. Okay, you get the idea. We execute, and it creates this grand neutral folder, and it adds these two averages to the preceding four. Okay, now I want to point out something else that has been made by the pipeline. This one here called Concatenated Average. Okay, so this one contains, for the selective attention case, contains all the averages that were generated up here, but they're put in one single file. Okay, so how are they coded? They're coded by subject ID and the condition. So this is for subject 00JJ up here, in the target ignore, in the selective attention, task conditions, and so forth. And all of, if we look at the events here, these you might call some kind of pseudo events, but the same mechanism works. So we have all these participants here, including the grand averages, and including what are called tags, which we can use to make averages. So we've got those there, and then similarly we have the same thing here under the neutral case. So okay, so what have we here? I made a new workspace that is simplified, because now we're paying attention to the averages and the grand averages. So this workspace has the concatenated averages that were generated for the neutral and the spatial attention. And then I merged them using a function of EMSE, and I saved the result. And here we see now what this one has, both the neutral and the spatial attention averages in it. It's got everything that, according to the experimental design. And so, in particular, here we're looking at in the neutral, the grand average for the target in the neutral case. Here we see the P3B. Indeed, it has a parietal distribution, and it's positive as expected. Okay, so I want to just sort of point that out. Okay, so now let's move on to the spatial component analysis. So what do we want to do here? We've got everything we want in this one file. So let's go to Analysis, Spatial Component Analysis, Infomax ICA, which is going to be equivalent to a front-end PCA followed by an Infomax rotation. We're going to use event-based segments. Okay, good. Now, we're going to make use of those tags that the event pipeline generated to make it a little easier. We just want things that are have a tag of grand or grand neutral. And our interval of interest, as I mentioned, was 0 to 800 milliseconds here. And let's go next. Okay, we don't need data acceptance rules. Everything has already been taken care of. We don't need to baseline correct again. We have nice baselines already. So let's proceed again. This question Do you want to use average instead of concatenated segments for PCA? The answer is almost always no. So let's say no here. Now, this is the front-end PCA part. So note here, if we take the first 4 components, they account for, these are accumulating power, they account for 98.1% variance from these 4. Okay, so let's look at the topographies of those 4. And so far we have 4 topographies which are labeled PCA1, PCA2, etc. And here they are, but these are constrained to be orthogonal, right? Which we as per our discussion, this may not be physiological. So let's proceed now to the ICA step. So I'll just say okay. Okay. This goes fast because we're working on averages and not raw data. And now here we have our 4 ICA components. And after we did PCA, we're just doing the ICA on those 4 PCA components. Now watch carefully here. These are still the PCA components. Now I click on Show Topographies. And now we're looking at the Infomax rotated PCA components, and you can see it made a difference, especially on these 2 components. Okay, I think I have 3 minutes left. So let me quickly go through the interpretation of these and see if we can give them the interpretation. So remember I said ICA1 was a P3B. Let's look here. So what have we done? We've taken the 128 channel averages and we've saved them as 4 channels, 4 ICA channels. That's down here in this concatenated one, and now we're looking at averages. Okay, so which one? Let's look in the target condition. Aha! So here we go. What are we looking at here? We see at 450 milliseconds in the target. The target is the black here, in the neutral, and this is negative. This is negative, so a negative times a negative gives us our positive. Okay, let's proceed to ICA2, which I called, for lack of a better name. It looks like we have a simultaneous anterior negativity around 200 milliseconds. It's simultaneous with a posterior positivity at the same latency. So let's look at it. So here we're seeing ICA2 here in the for the standards, and here we see in the 3 cases the ignore condition is in magenta. It's the one that shows the largest. But let's look at the targets. And let's see, here we are in the targets, and we can see here there's a differentiation. The ignore is now larger, so maybe possible interpretation. The subject has to ignore, suppress. So even though this is a standard, perhaps, oh no, these are the targets. So maybe this has something to do with suppressing, or the effort required to ignore the Okay, I think I'm just out of time, but let me quickly point out that I think ICA4 is going to be the more. I didn't say ICA3 yet, but let's jump ahead to ICA4. This one shows a differentiation here at 385 milliseconds between there's a polarity reversal between the target ignore in magenta and the target attend in blue, and the neutral condition is right in between. So that's potentially interesting. And as we look at the picture of it, potentially right inferior frontal cortex has been shown by some authors here to possibly be related to inhibition or putting on the brakes, which might be the case here since we're doing suppression. Okay, finally, I know I'm out of time, but let me say one quick word about ICA3, which I called motor. It's plausible that these contour lines could be consistent with the motor strip, but let's look at the waveforms. Here, we can see it clearly for ICA3 in the target ignore condition, which is magenta. Participant is correctly withholding a response, and we see how flat that is. Whereas in the target neutral, when the participant always responds, and in the target attend, when they also respond, we see here. So this is where I think the response time distribution is occurring. Okay, so there we have it. Thanks for your attention, and have a great meeting.